Hello guys, welcome to my channel. Today we'll discuss about the another part of second chapter, that is sexual reproduction in flowering plant. So today's topic is pre-fertilizations and structures and events of pre-fertilization. So what is pre-fertilization? Pre-fertilization is the process that occurs before the actual fertilization. That means the events that occurs before the fusion of male gamete and female gamete is known as pre-fertilization. Pre means previous. Pre-fertilization is the process that occurs before the actual fertilization, that is the process before the fusion of gametes. There are two events that occur before fertilization, that is gametogenesis and gamete transfer. So what is gametogenesis? Gametogenesis means formation of male and female gametes, and gamete transfer means bringing the male and female gametes together. So what is gametogenesis? It is a process of formation of gametes. Male gametes are formed inside the anther and the female gametes are formed inside the ovule. That means gametogenesis means the process of formation of gametes. Now the structure of gametogenesis. Structures that let us have a look at the details of the structures involved in the process of gametogenesis. That means the structures which helps in gametogenesis we will discuss now. The first structure is megasporangium. Megasporangium or pollen sac. It is a sporangial structure surrounded by four walls, that is epidermis, endothecum, middle layers and tapetum. So all these structures we discussed previously. If you don't see the video, you can see the video in my channel. So the pollen grains are formed in this. There are four pollen sacs. The center of each sac comprises of a group of exactly arranged homogeneous tissue called sporogenous tissue. So megasporangium is a four-layered structure, that is epidermis, endothecum, middle layers and tapetum. Here pollen grains are formed and four pollen sacs are present here. Another structure that involves is ovules. These are the place for the formation of female gametes. That means female gametes are formed inside the ovules. These are present at the bottom of the vase-like structure called carpel, which has a neck called style and the opening called stigma at the top. Here this is a structure of mature ovule. And another is pollen grains. Pollen grain is a microscopic structure that represent the male gametophytes. That means pollen grains represent the male gametophytes. Pollen grains are usually 25 to 50 micrometers in diameter. It has a prominent two layers wall, exine and intine. That means the outer layer of pollen grains is known as exine and inner layer is known as intine. Here this is a enlarged view of pollen grains. Four pollen sacs are present inside the pollen grains. These are pollen sac. Anther. It is a bilobed structure. The two lobes are attached to the stripe of the sterile part. Here anther is a part of stamen also. This bilobed structure is anther. And another structure is stamen. It consists of a stalk called the filament. The proximal end of the filament is connected to the petal of the flower known as the thalamus. Here this stalk-like structure is called filament and this structure is thalamus, and here this part is anther, which is attached with the stripe of the sterile part. Now the formation of male gametes. That means how male gametes formed during pre-fertilizations. Now the male gametes formed due to two ways, that is microsporogenesis and microgametogenesis. Microsporogenesis means how male spore are formed. Microsporangenesis is the initial process and marks the development of microspore or pollen grains in the pollen sacs of anther, known as microsporangenesis. That means how pollen grains are developed from pollen sac is known as microsporogenesis. The diploid pollen mother cells gives rise to 4 haploid microspores by meiosis. That means diploid pollen mother cell gives rise to 4 haploid microspore by the process of meiosis or meiosis cell divisions. Each microspore develops into a pollen grain. Here 4 haploid microspores, after development it becomes a pollen grain. Now the microgametogenesis, that's mean formation of male gametes. Formation of male gametophyte from the pollen grain is known as microgametogenesis. Pollen grain undergoes mitosis and gives rise to 2 cells, small generative and large vegetative cells. Pollination takes place here. About the generative cells and vegetative cells we discussed in our previous video. The generative cells give rise to 2 male gametes, while vegetative cells give rise to pollen tube. That means generative cell, it becomes the male gametes, and the vegetative cells, after development it becomes pollen tube. Now the formation of female gametophytes. That means how female gamete produce. Here female gametes also produce due to 2 ways, that is megasporogenesis and megagametogenesis. What is megasporogenesis? The megaspores develops inside the megasporangium. This is known as megasporogenesis. That means the development of megaspores or female gametes is known as megasporangenesis. The process takes place inside the ovule within the structure called nucellus. It means female gametes are produced inside the ovule. The diploid megasporocyte inside the ovule undergoes meiosis to form 4 haploid megaspores, and only 1 megaspore develops into the megagametophyte while the other 3 disintegrate. That means diploid microspores, under the meiosis cell division it become 4 haploid megaspores, but out of these 4 megaspores only 1 is developed into megaspore and other 3 is destroyed or disintegrated. Now the megagametogenesis. The female gametes form from the functional megaspore. This is known as megagametogenesis. That means productions of female gametes from female functional megaspore is known as megagametogenesis. The megaspore undergoes mitosis thrice and rise to an embryo sac within 8 nuclei. Each micropyle and chalazal end contains 4 nuclei initially. At the time of fertilization, one of them from each pole fuse to center and form a diploid nucleus. The 3 cells at the chalazal end disintegrate later. At the micropyle, one of the 3 cells develops to form egg cells. The remaining 2 secrete chemicals to guide the pollen sacs. That means out of 4 cells, the 3 cells develops to form egg cells and 1 is secrete some chemicals which guide the pollen tube. Now gamete transfer. Gamete transfer means how male gametes transfer into the female gametes, or how male gametes and female gametes come contact to each other. Formation of seeds requires pollination. In this process, the pollen grains from the anther are transferred to the stigma. Gamete transfer is also known as a pollination. That means how pollen grains transfer from one flower to another flower, or the male anther transfer to the stigma of female flower. Pollen grains germinated on the stigma and reach the ovule through the pollen tube. And the male gametes are then released near the egg. How gamete transfer? This process we had discussed in the pollinations. These are the whole process that occurs in the pre-fertilizations of a flowering plant.